get started then. So first of all, thanks very much, Diane, for the invitation to allow us to present our project here. Duchenne is a new indication for Pfizer, and what I want to talk to you about today is a phase 2 clinical study that is in the setup phase, and that will be open in the UK in the first quarter next year. So Pfizer, I've recently moved to Pfizer, and I was drawn there because of the establishment of a rare disease discovery unit. Big Pharma have not traditionally been very active in rare diseases, but clearly Pfizer made a commitment not only for the repositioning of their drugs into rare disease indications, but also to do new drug discovery to address high unmet need in rare diseases. And our particular focus is on neuromuscular disease and rare hematological disease. Before I say any more, I have to make this disclaimer. So what I want to talk to you about is a little bit about a background of myostatin biology, some of the potential approaches to interfering with that biology, and some background on a monoclonal antibody that Pfizer are progressing into phase 2. We have some data from preclinical studies in mice and monkeys, and we have some data from a phase 1 study in healthy adult human volunteers. So this is the target that we're trying to interfere with. It was identified as being of pretty profound physiological importance about 15 years ago in the late 90s. Through the analysis of spontaneous mutant cattle and targeted genetic deletions in mice. And the phenotype that results from an abnormality in the production of myostatin protein, which is a small peptide hormone, is of increase in muscle bulk and increase in muscle strength. And it turns out that this has been highly conserved in evolution. So every species that's been examined has shown this, this same genotype to phenotype correlation. And indeed in man, there have been some reports of a few children born with a deficient myostatin gene who have a phenotype of increased muscle bulk and increased muscle strength. Importantly, there's no impact of these genetic mutations on lifespan or apparently on any other organ other than skeletal muscle. So the protein is produced as a propeptide. It's produced by muscle cells, and it signals to muscle cells. So it's what we call an autocrine hormone. It's produced with it's translated with a propeptide, which within the muscle cell is cleaved to form this myostatin latent complex made up of the propeptide and mature myostatin protein. This is secreted and released into the circulation and is inactive in this form when it's complexed with the propeptide. Undergoing proteolytic cleavage, the native hormone is exposed and can interact with this receptor, the activin R2b type receptor on the surface of muscle cells, and induces some downstream signaling, most notably with the phosphorylation of SMAD transcription factors. So with this genotype to phenotype correlation and our understanding of the molecular biology, we can obviously hypothesize that there may be ways to intervene with this biological process by complexing this native hormone. So in the subsequent decade, a fair amount of further cellular biology information has emerged to suggest that the action of myostatin occurs both on very early muscle progenitor cells and in the differentiation and maturation of myoblasts into myotubes. So it acts at multiple points on in the process of differentiation of an activated muscle satellite cell into a myotube. And it didn't take too long. You'll notice 1997 the gene was identified and published, and by 2002 there were already publications looking at the interaction of myostatin biology with the Duchenne genotype. So most notably in studies where genetic knockout mutants were crossed, the MDX animals and a myostatin knockout animal were crossed. And it gave the opportunity to look at the development of muscle in the MDX animals in the absence of myostatin. And while it's clear that this is not normal muscle, it's also clear that there is a reduction in the degree of fibrosis and fat replacement that occurs normally in the MDX muscle by that stage. Similar improvements in the development of muscle were noted when myostatin was complexed with an antibody and neutralized that way. There were also some improvements in the quality of muscle. Investigators strayed into dogs, and most notably with the delivery of an adenovirus that delivers a myostatin inhibitory signal. Similarly, this is the golden retriever model, a reduction in the degree of fibrosis and adipose tissue replacement is seen in the treated animals after a long period of treatment. As we're on the theme of dogs, so it's worth pointing out that this bully whippet, which was the dog strain that was first identified as having a myostatin mutation, performs very well as a racing dog. And in a study of a population of racing dogs, it was clear that some of the best animals were either heterozygotes for the mutant myostatin gene or homozygotes. And this group was very much overpopulated in a racing dog population. And the reason I point that out is that it may indicate that there's a gene dosage effect, that you may not need to completely neutralize myostatin too. So we heard yesterday from Kay just how difficult drug discovery and early drug development can be. And I wanted to highlight that from the demonstration of neutralization of myostatin in MDX animals in 2002, there have been a number of programs developing in muscular dystrophies to look at myostatin neutralization. The first was a study of Myo29. This was a Wyeth antibody, and it was studied in a range of muscular dystrophies, Becker's and facioscapulomuscular dystrophy, and others. primarily for safety, but also looking at muscle strength. The development was stopped ultimately because of an apparent lack of efficacy, at least at the doses that were well tolerated. So at higher doses, there was a fairly frequent occurrence of a skin rash. Secondly, there was a study conducted by Acceleron Pharma in partnership with Shire of a drug called ACE-031. And this looked very promising, this study. But in fact, again, the development was stopped because of a safety signal. The safety issues, as far as we can understand, were related to nosebleeds and the formation of telangiectasia in the skin, which are little vascular abnormalities in the skin. Now, it turns out that this inhibitor is actually a protein of the receptor for myostatin and has a much broader ligand specificity than some of the antibodies, and particularly the antibody that we're progressing. So one of the genotype abnormalities of hereditary hemorrhagic telangiectasia, a genetic condition where you get nosebleeds and telangiectasia in the skin, is a defect in GDF9, closely related but irrelevant for the purposes of muscle biology, binding partner of this drug. So when Wyeth and subsequently Pfizer started looking for improvements of antibodies that we could again progress into muscular dystrophy, was taking into account these two past failures. So first of all, we needed to find an antibody that neutralized GDF8 that had higher potency and greater efficacy than the previous antibody MYO-029. And secondly, we had to be very sure that our antibody did not bind to GDF9, which caused these nosebleeds and telangiectasia. So we screened many antibodies from the Wyeth stable, and I've identified one that certainly does not bind GDF9. And in functional testing in mice, in normal mice this is, we could demonstrate not only good potency but a greater level of efficacy on a functional muscle measure, that of tetanic force generated in the mice. And subsequently, to build our confidence, we've seen a dose-response relationship in cynomolgus monkeys. We're dosed for a period of about 8 weeks, and when we measure muscle increase by MRI scanning. That's given us a lot of confidence to move forward. Furthermore, we were able to show that after 8 weeks of dosing, there was a prolonged benefit in terms of lean body mass, or lean leg mass in this case, in primate species. So we have a spontaneous mutation of GDF8, myostatin, and its deficiency, which is associated with an increase in muscle mass and muscle strength. We have targeted knockout animals and therapeutic inhibition approaches that produce a very similar phenotype. In myostatin deficient dogs, there are clearly functional improvements in performance and in vitro evidence of increased muscle regeneration rate associated with myostatin inhibition. Finally, looking at dystrophic models in both mouse and dog, we've seen that the mechanism can ameliorate therapeutic myostatin inhibition. For our antibody, this one, we've repeated the studies in the MDX mouse and confirmed that delay and protection of muscle damage. So this antibody went into phase 1 testing that's now complete. We're in the process of reporting the study at the moment. And this study looked at a dose range of 1 to 40 milligrams per kilogram dosed intravenously on one occasion, and a dose of 3 milligrams per kilogram dosed subcutaneously. And in addition to that, these volunteers were dosed with 10 milligrams per kilogram on 3 occasions 2 weeks apart. So we could say from that experience that there were no safety issues, that the drug was generally well tolerated. The drug behaved from a pharmacokinetic and pharmacodynamic point of view as we'd expect it to. We had pre-specified a proof of mechanism of an anabolic effect as measured by DEXA scanning, but in fact the variability was rather higher than we had expected, and we did not achieve that proof of mechanism. But in the human volunteer study, we also looked at several MRI measurements, and I'll show you some of those, which suggest to us that there is some real pharmacology with this drug, even at the detectable in small numbers and at short periods of dosing. So this is the pharmacokinetic profile. It's linear and has our half-life typical of IgG1 monoclonal antibodies. And over here on this side, it's slightly confusing to people. This is a measure that we use to show that the antibody is actually binding the target. What happens when it binds the target is that the short half-life of GDF8 is prolonged and becomes the half-life of immunoglobulin, and so it appears to increase in concentration in the circulation. But it's inactive because it's bound to the antibody. So this gives us confidence that in vivo, in these volunteers, we've neutralized GDF8 in the circulation. When we looked at lean body mass by DEXA scanning, there was a trend, if anything, it certainly wasn't statistically significant, of an increase in lean body mass as you increase dose. But most compelling really were when we looked in that group of patients who received 10 milligrams of the drug 2 weeks apart over 1 month. Dosing has stopped here, and in the subsequent 3 months after the cessation of dosing, it was possible to measure statistically significant increases in the percentage of probable muscle as measured by MRI in the T2. We also saw evidence of an increased muscle volume at 4 months as measured by T1 MRI. So those MRI measures have given us the confidence in the context of a safe drug in phase 1 testing to move forward into phase 2 evaluation. And here's just the summary. So we met our PK and PD modulation that we expected and had some evidence of pharmacology. So what we propose to do subsequently is to move into a phase 2 study that I'll tell you about in a moment. Patients who have completed that study will be eligible for an open label extension study. And then we anticipate some sort of phase 3 study. We don't know whether that's going to need to be a confirmatory study in young boys, age 6 to 10, or a study in more advanced disease. And we'll decide on that depending on the outcome of this study. So the study that we're proposing is a randomized placebo-controlled study. We're giving the drug over a 2-hour intravenous infusion every 4 weeks. There'll be about 105 boys recruited to the study in the age range of 6 to 10. They'll need to be ambulatory and on a stable dose of glucocorticoids. We're asking for some impairment of the four-stair climb prior to, as part of the screening evaluation. There'll be an individualized dose escalation. So after 4 monthly treatments of drug, there'll be a safety review for each individual subject. And if they satisfy that set of safety criteria, they'll be allowed to escalate to the next highest dose. If there are any concerns, they can stay on the same dose. If there's a dose to reduce to, they can reduce. Or, of course, the drug could be stopped. At every 4-month time point, there'll be an MRI scan of muscle, looking at T1 parameters for muscle volume, T2 parameters for muscle quality, and if we can acquire the sequences, the Dixon set of evaluation. That will be performed every 4 months. Now, our primary endpoint for the study, we've selected the four-stair climb. This has caused some interest and attention. I think I should tell you why we've done that. So first of all, our mechanism of action is primarily, we believe, will be to increase proximal muscle strength. And the four-stair climb is probably a better reflection of a clinical measure that correlates well with muscle strength. Secondly, in our analysis of the natural history data, which the community, in particular the Synergy group, has generated, we believe that the four-stair climb is, in prospective studies, a good deal less variable than the 6-minute walk distance. So it allows us to statistically power a study with a smaller sample size. Thirdly, the steroid studies that we heard about earlier on today from the late 80s and published in the early 90s clearly described a statistically significant change in the four-stair climb within 6 months. And that the size of that effect gives us a benchmark with which we can compare our drug to some extent. And so those were the major reasons that we've selected the four-stair climb as the primary endpoint. We will be looking at other functional endpoints, the North Star set and the 6-minute walk distance. But for our statistical purposes of the comparison for a primary endpoint, the four-stair climb is best suited for this study, which for us is a proof of concept study to demonstrate that we have a real pharmacological effect. We need to choose the endpoint that is best suited for that purpose. So after a 48-week period of treatment, the boys will go on to a second period. Those who receive placebo for the first 48 weeks, let's call it 12 months, will then go on to this dose escalation regime. Those who are in this randomized to this treatment group will continue on their maximum tolerated dose of the compound. And those in this second sequence will not be re-randomized, but they'll have been randomized to this sequence at the baseline, will go on to receive placebo. Now, the importance of this is that it allows us to see how long any effect is maintained after we stop treatment. We need to study the pharmacokinetics after repeat dosing in this population, and we need to look at the immunogenicity of our drug. And so for those reasons, this group is critically important to the study to allow us to get that information. So although it may seem inappropriate if we, for example, were to have a positive primary analysis that we have people on placebo, in fact, because of the half-life of the drug, there will be drug around for probably 2/3 of this period. As I say, it's very important that we're able to provide evidence to the regulators of the pharmacokinetics, the decline of any benefit, and the immunogenicity of the drug. So our primary analysis will be after a year. We'll continue, boys randomizing into the study will stay on the study for the second year, and they may receive continuous long-term treatment out to the second year on their dose, or if they were randomized to placebo in the first instance, will receive active compound. Of course, we'll have an external data monitoring committee who will be reviewing safety data in particular, but also efficacy data on a quarterly basis. So, I hope I pointed out to you that from the point of view of the biology, there's a critical role of myostatin in the regulation of muscle growth. That inhibition of myostatin by multiple modalities in several different species has resulted in increased muscle mass and can lead to increased performance. That in animal models, myostatin inhibition seems to ameliorate disease, and that we have an IgG1 monoclonal antibody that is progressing through phase 1. I've shown you some of that data. We're currently having regulatory meetings with 5 regulatory authorities. I think we're on the last one next week. And we have a phase 2 protocol that I've described to you and I'm happy to answer questions on that is being set up according to our timelines, and in fact our first site initiation is next week. It will be conducted in the US, Japan, Canada, the UK, and we hope in Italy. So I've realized I've rattled through that rather faster than I'd intended, but it leaves plenty of time for questions and means that you can get slightly early to lunch, I hope. I'll leave it there.
Thank you, Michael. Yeah, we've got about 15 minutes for questions, so I'll take that.
I was wondering whether in human, in DMD, myostatin is important, you know, the myostatin axis, the receptor, or the peptide itself. Is there any evidence that it's a major root of muscle degeneration?
So I think the most pertinent piece of data we have has been an analysis of myostatin levels in Duchenne boys compared with a normal population. And what I can say is that it's not elevated, and if anything, the levels are lower than in a normal population. What you've got to remember, of course, is that muscle is the primary tissue of production of myostatin. And what we're measuring are circulating blood levels of the hormone, and not local levels. Unfortunately, we don't have good assays of downstream GDF8 signaling that will stand up to clinical interrogation. So there's a bit of a black box there. That particular analysis doesn't give you a nice warm feeling that it's elevated and therefore if you take it away then it may have an effect. But I think our only comfort there comes from the preclinical disease models in the MDX and the golden retriever model. So from clinical studies, I don't think we have that. Does that answer your question?
I had two related questions. Thank you very much for your presentation. The phase 2 trial seems very well powered. I wondered whether you, in your discussions with the regulators, were talking about taking advantage of their wonderful accelerated approval schemes to allow patients earlier access to medicines. And secondly, whether you had in your discussions considered using natural history data and the North Star data instead of placebo.
Okay, so first of all, I have to be a little bit careful what I say, but yes, we have addressed the possibility of accelerated approval on the basis of either the primary or the secondary analysis here, but most importantly the primary analysis. And I think I can fairly say there's an openness to look at that if we generate a statistically significant signal. So on the basis of the 4-stair climb, some of the regulators wanted more supporting data, good trends in the same direction of other endpoints. Some were less concerned about that. So I think what we've seen is an increase in the flexibility of some of the regulators to looking at other clinical endpoints that may not be directly a demonstration of benefit on quality of life, which is what they would all ideally like to see, but a functional clinical measure that can clearly be interpreted as of benefit. And I think perhaps what helps us here is that the data from the steroid studies on this endpoint showing the magnitude of effect and clearly over long-term observation these have been adopted as standard of care, if you like, for the treatment of this disease. So, you know, we're not coming to this endpoint from a vacuum. There's 25, 30-year history of neurologists using the 4-stair climb endpoint. We believe it's more relevant to our mechanism of action, and we believe it's less variable. And we've seen flexibility with the regulators to look at that. Obviously, they'll never commit, but there's certainly flexibility.
About using natural history rather than placebo, especially if you have this natural history data on the 4-stair climb.
The difficulty is that the populations are always changing, and the particular population that we look at is quite difficult to pick out from natural history sources. I think we took the view internally that to have confidence to move forward, we'd want to see activity against placebo, just because of all the attendant risks of interpretation of that kind of comparison. So we didn't take that question to the regulators.
a situation where you would take that to the regulators? I mean, we're talking about an old paradigm of drug development where placebo-controlled trials are absolutely fundamental. But in rare disease populations where the population eligible for clinical trial is dwindling and every month of a young boy with Duchenne's life is crucial in terms of muscle deterioration, can you see a situation in the future where there would be an acceptance that placebo-controlled large-scale trials are unethical?
Well, I think it depends on the duration period of treatment, partly. So, you know, if you do have long observation periods and there is incontrovertible natural history data to support the magnitude of the decline, and that you believe that you can power against that, then absolutely. I don't see why that should be an issue. We didn't feel that we were going to be in that position with this study, and so we've opted to keep a placebo in the study. The important thing from our study design, though, as well as trying to be as economical as possible, is to ensure that everybody going into the study does get a 12-month period of active drug.
So given the need for a placebo, as you've stated, for this study, after the end of the second period, will there be plans to allow boys to extend on the drug?
Right. So we are planning an open-label extension study that will be available for those boys as they emerge from period 2. Exactly what that study looks like, we don't quite know yet. It is going to depend on data that emerges from the primary analysis, but the plan is to have a study in place for continued therapy.
Thank you. I've got a couple of questions. What are your key secondary endpoints? And secondly, would you see this as an add-on to other drugs for DMD, or do you see this as a standalone drug?
So in terms of secondary endpoints, we are looking at the 6-minute walk distance. We are looking at the North Star assessments. We are looking at other exploratory endpoints. And we are looking at MRI. And so MRI is performed at 4 monthly intervals at each of those dose groups, dose escalation periods. So we will be generating that data. Yeah.
And the other question?
So we're already considering it as an add-on to glucocorticoids. We've set that in the study. For the future, we don't see any reason not to consider an add-on. For this study, clearly we need to keep it homogeneous. Certainly no obvious contraindication to combination.
Any further questions?
I have one more.
Sorry, I think I might have missed that, but in your new study you mentioned that the age range between 6 and 10. What was the age range in the study before that?
With the Acceleron study?
Yes, that's right.
Sure, I can remember. I have to look it up and get back
Would
to
it
you.
have been quite similar?
I think it was probably wider. It was wider. There you go.
Oh, 6 to 15. And I was wondering if there were any significant changes that were seen or you expect to see between changing the age range in terms of your antibody binding profile.
In terms of the binding profile?
Yeah, in terms of just how effective it is.
Well, we wouldn't anticipate that. Mm-hmm. The reason we've restricted the age range, as you've heard from multiple investigators and companies, is for homogeneity on the endpoint. But from a biological perspective, there isn't a reason to think that this could not be effective in adolescence or in adulthood to increase muscle strength.
Great, thank you.
We have to prove the principle somewhere.
Yes, of course. Thank you very much.
Hi, it's just a question about the forced stair climb as the primary endpoint. I was just wondering if anybody considered that recent research has shown that actually it's a lack of ankle range of movement that hinders the boys going up the stairs initially. So it might be that instead of capturing a decrease in muscle strength, what you're actually capturing is an increase in contracture, because as soon as the boys lose past 90 degrees, they then start to have difficulties no matter what strength they've got.
So it's a great point, and we will be measuring ankle range of movement through the study. So if there is a change in that measurement, we'll be able to allow for it.
Will there be a limitation on ankle range in the inclusion criteria as well?
Not that I can recall.
Okay.
I should say that the study will be going live in clinicaltrials.gov next week, we hope. By the end of next week, we're told. And the key inclusion-exclusion criteria, in fact all the inclusion-exclusion criteria will be listed there.
I'm afraid